Welcome everybody that is also in the other room. Welcome to this second seminar of these conversations between ISCIII and IARC. And this is the first one. It's going to be more like no more. It's a scientific one. I just want to remind everybody more or less the structure of this seminar, which is like two small seminars, one from people from IARC and another seminar from people here at ISCIII or from our friends of ISCIII. And today I'm gonna let Ana Gonzalez be the chairman. She is the head of the Human Genotyping Unit at CNAO, and she knows much more than me about all these types of the subjects. So Ana, I give the floor to you.
All right. So welcome everybody to the next conversation between Instituto de Salud Carlos III and International Agency for Research on Cancer. So the title of this session is The Impact of Genomics in Disease Prevention and Treatment. And we have for this conversation two amazing speakers, Professor Ángel Carracedo and Professor Paul Brenner. Also is going to talk the Dr. Lynette Fernandez Cuesta and Sandra Perdomo from the IARC. And then after the talk, we will have time for discussion and questions. So we hope you enjoy it. So let's move for the first speaker. It's been the Dr. Ángel Carracedo. Let me introduce him very briefly. He's the director of the Galician Foundation of Genome Medicine, director of Genomics and Bioinformatics Group in CIMUS in Santiago de Compostela, director of the Spanish National Genotyping Center in Santiago de Compostela, director of Translational Medicine Group in CIBERER, professor of legal medicine, and president of the International Academy of Legal Medicine. So his career activity started in population and forensic genetics, being a pioneer in the use of new technologies in forensic identification, and being the director of the Instituto de Medicina or Institute of Legal Medicine during 20 years. In 2002, he also started working in clinical genetics area, and he has set up the Galician Foundation of Genomic Medicine. He has already published over 800 international scientific papers and 8 books. He is currently coordinating the impact program in genomic medicine and the mirror group in the 1 Million Genomes Project, been participating to date in more than 20 international projects, coordinating 7 of them. Current president of International Academy of Legal Medicine, board member external advisors of different societies of forensic genetics, cancer, and pharmacogenomics. He has received many prizes at distinction and as JAMA First Award and National Award of Genetics. And he is Doctor honoris causa for 8 universities in Europe and in America. So Ángel, thanks for the participation of this event, and we will look forward to hear your presentation titled Translation of Genomic Medicine to Clinics, Challenges and Opportunities.
Thank you, Anna, for the nice introduction. Hello, everyone. And thank you for the invitation. And thank you especially for the presentation, Anna. Too kind. But anyway, I think that I am geneticist. I am very proud of coordinating quite a large group of people dealing with genomics and application of genomics in general terms, but very much focused to applied genetics. I'm going to give in half an hour, I hope, a general view of the problems of translation of personalized medicine. Personalized medicine nowadays is mainly genomic medicine, although in the future maybe it will be different. And I'm going to talk about also the IMPACT project. This is this initiative that we have in Spain for the implementation of the genomic medicine in the health system. And let me start with a definition of personalized medicine. The idea of personalized medicine is targeted treatments for a reduced group of patients to improve efficiency and to reduce side effects. For this, we need biomarkers And then we need diagnosis, but it's also early detection of patients at risk. Improving preventive measures, usually individual or in groups of people. Personalized medicine is going to be more and more expensive. And then the only way that the social health system has is to invest in prevention. I very much persuade on that. And but now the promotion of personalized medicine, and we are talking about precision and personalized medicine all day long. It doesn't come from the academy, but from the industry. And the industry is because the industry has a problem. The cost of production of a drug has been reducing more or less at 20% in the last 10 years, but also the productivity. It's more and more difficult to have a drug approved by the regulatory agencies. And the problem also is that in the old times, they usually fall in the first steps due to unpredictable pharmacokinetics. But now the chemistries, they are able to modify everything and to make any type of molecule druggable. And the problem now is efficiency. We have reached a plateau of more or less 58% of efficiency that is extremely difficult to improve. And this is a problem because if the drug is not more efficient than what we have in the market, is not approved by the regulatory agency. And after a very, very large investment, this has catastrophic consequences for the companies. And that's why, and we knew that, the idea is to identify groups of people, link it to biomarkers where the drug can be more efficient, so they can save the product, the molecule, at the end of the day. But there is also another problem with the drugs, is that the number of advisory drug reactions, they represent 5% of the hospital admissions in Europe, is between the 4th and the 6th cause of death. There are more people dying by direct ADRs than for traffic accidents, for instance. And the reason for this is what we call, many years ago, the paradox of the modern drug development. Clinical trials provide evidence of efficacy and safety in populations, but the physicians treat individual patients who can have different response to drug therapy. For instance, my father could take an anti-inflammatory drug without any problem, but I need to take some antacids, otherwise I have problems. And this is because we have individual variation. But the main problem of the lack of efficiency is the heterogeneity of the disease. And disease heterogeneity is the norm, obviously for complex diseases, but also even for Mendelian diseases. And unless we are able to stratify the disease in groups where the drugs are more efficient, and this should be usually linked to the ethiopathogenical mechanisms of the disease, it's going to be impossible to have more efficacy. So we need to stratify disease, and for this we need biomarkers, and also we need to identify biomarkers for drug response. And this is more or less the basis of the personalized medicine. This is complex, it's very heterogeneous, and any disease has a genetic component and environmental component, and the drug response in an individual can be different, and this is also genetic and environmental provoked. In the genetic part of the second part of the safety, one important thing is that many of these different drug response are these advisory reactions are related with metabolic genes, especially CYPs, and there are very, very important population differences on that. But anyway, we need we need biomarkers, and most of the biomarkers we have nowadays for for personalized medicine come from genomics. And all the whole the different steps of the genomic biomarker for personalized medicine involve 3 different steps. One is discovery, is research. Second is validation, and in the case of the drugs, regulatory approval. And the third is translation. If we are able to identify with research biomarker, is approved by, imagine, by the European Medicine Agency, and is in the clinical seat of the drug, but we can't translate this to the clinical settings, we have a problem as well. Concerning the phase of discovery, perhaps we have 2 main strategies for identification of biomarkers. First one is next generation sequencing, more properly called massive parallel sequencing. Everybody knows this in this context, so we don't have to talk about this very much, but just to mention that the equipment, the system has an incredible fast evolution rate with that means a high equipment renewal rate. It's not only sequencing, but we need a computing storage capacity and bioinformatics as well. And perhaps the most important impact in personalized medicine and diagnosis of disease are in 2 areas. One is in rare diseases, and the second one is in cancer. The third one is pharmacogenetics. Obviously, next generation sequencing revolutionized the diagnosis of the Mendelian genetic disease. You know that ISCIII is the International Rare Disease Research Consortium. This is a huge initiative involving most of the countries in the world. It was launched something like 20 years ago. I know, not 20, 14. I am this one here in the left when I was young, but it's yeah, I was young. It's only 12 years. That's quite shocking. I was more younger then than I am now. But anyway, we have always goals in the ISCIII for trying to get some objectives from every 5-10 years. Now the expectation of the ISCIII is to have a diagnosis of the 90% of the rare diseases within 1 year from symptoms in 2027. And I think this goal, despite the use of next generation sequencing, is very ambitious, but is difficult in Spain, at least, that we are going to reach this goal. We have in this moment an average of 3.6 years. And Spain, I think, is going to be difficult, and not because of the sequencing capacity, not because the bioinformatic, but because the lack of clinical geneticists and perhaps the organization of the health system in this given area. I think it's more possible to get a number of treatments for rare diseases. We have more or less 150 orphan drugs approved so far for the European Medicine Agency, expected to have around 1,000 in 2027. But I think we are able to get this because the improvements of gene and cell therapies and because of the big pharma has also entered into the market. I mean, they are very, very interested in rare diseases. So I'm pretty sure we are going to get this objective. But one of the problems is that we are, one of the consequences of personalized medicine is that, and the stratification of disease, is that we are we have a shift from common disease to rare diseases. The number of rare diseases is continuously increasing every year after year. And it's because of the stratification of the disease with a high genetic component. Imagine the case of the autistic spectrum disorders, of the intellectual disability. They are very, very common disorders. ASD, autistic spectrum disorders, is less than 1 in 100 people, newborns. Intellectual disability, 1 in 50. But now there are more than 300 rare diseases, and we are able to reach diagnosis in more than 30% of the cases of autism and more than 40% of the cases from intellectual disability. And more and more, these diseases are going to have personalized treatments and extremely high cost. So I think that we need to make a reflection on this, because the problem is the sustainability of the national health systems. As Anna mentioned, I am the director of here in Galicia is an autonomous region in the northwest of Spain, and here everything, all genomic diagnosis is concentrated in a lab of the with the legal structure of a foundation of the Galician health system, and we have obviously an incredible amount of work. Half is cancer, half are rare diseases, and the movement is we are changing. Now we are using for everything whole exome sequencing for the diagnosis of rare disease. We have moved from gene panels to whole exome sequencing. Maybe in the future, everything will be whole genome sequencing, very, very likely. But the bottleneck is not sequences, it's not bioinformatics. The bottleneck is when we have the variants prioritized to the report. The data mining still doesn't work. And then I think this is a big problem, because it's difficult to give a solution to the patients in a fast time with the public resources, unless we are able to reduce the time we need for this. We think that perhaps a strategy is to train the clinicians to use human phenotype ontologies, and we are doing an effort on that, a very, very important effort on that in Spain. The second field that has had a big huge benefits of the next generation sequencing is cancer, that you are going to talk about this in the next speeches. But we were involved in the International Cancer Genome Consortium in the Spanish effort, and I think that all this international progress in cancer that allows us to see the landscape of mutation and to identify the drivers, link it to the advances in monoclonal antibodies, allow for targeted therapies that have improved a lot survival. And much of the work, 50% of the work we have is related with this, is with identification, with targeted therapies, and identification of mutations for cancer. But for common disease in general, common disease in general are caused, common disease is not caused in general terms, and not due to rare functional variation, but for common variation. And many genes that contribute to the at low odds ratios to the disease. And for this, what we need is association studies, especially genome-wide association studies. And this type of studies have, with this type of studies, most of the biomarkers that we have nowadays for the stratification of common disease, for instance, schizophrenia, bipolar disorder, everything has been identified using GWAS. And but the same for the biomarkers used for ADRs. Most of them have been also identified using GWAS study. As Anna mentioned, we have in Spain the National Genotyping Center. Anna is coordinating one in the nodes in the CNU in Madrid, and we have another node here in Santiago. Now we're going to talk about challenges very, very briefly. The challenge we have in personalized medicine in general terms, in translation first, is the validation. Validation is mainly performed by the regulatory agencies. I am very proud to have helped to the implementation of the pharmacogenomic working group in the European Medicine Agency. In the FDI, they have a similar group, more active than the EMA group. they have more money and perhaps a better organization. And but we have defined a general biomarker qualification process between the two regulatory agencies. And in this moment, just to mention that around 50% of the drugs, exactly something like 900 drugs, they have biomarkers that sometimes are considered to be required, that's mandatory, recommended, actionable, or informative. Especially the first two are mainly mandatory. And they have indications, even sometimes those recommendations in the technical sheets. If you want to see the status of the different drugs, the best way to do it is using the web page of the American Consortium, the FARC-GKB, where you can see the status by active principles of for the main agencies. And obviously, especially in cancer, some of the valid biomarkers are widely used. But I have the impression that we are being guided by the industry too much. And some things that they have that could be translated to the clinical settings are not still, if they are not promoted by the commercial companies, by the pharma companies, they are not still used. But obviously, in many cases, we are using biomarkers, especially in cancer, but not always. Imagine that, for instance, at least in Spain, in a first year of colorectal cancer, we usually use a combination of called FOLFOX of 5-fluorouracil, 5-fluorouracil and oxaliplatin, or 5-fluorouracil or capecitabine, and irinotecan. And there is a mandatory drug for irinotecan. There are variants of in UGT1. That means 1 to 2% of the population. They have a strong problem of medulloplasia, of granulocytosis, if this is not performed. And it's very, very rare that this analysis is requested to the labs. It happened to say to the DPID in the past. Now in Spain, because of the Spanish Medicine Agency, they promoted that they should before giving capecitabine of 5-fluorouracil, DPID testing is mandatory. Only in this moment we have lots of cases, but not before. The same is true for somatic mutation. We need still to progress in validation schemes, and especially in Spain, we need to agree with a portfolio of services of our national health system, because because in this moment it's quite chaotic. Different labs using different rules, different biomarkers, and that's something that we need to progress as well. But if the situation in cancer is not bad, in other medical specialties is more complicated. For instance, psychiatry. This is one of the specialties that could have more benefits of biomarkers and personalized medicine. But I mean, they are not so well guided as oncologists. So we need to education, and we need to promote this. In Galicia, we have started a strategy to educate and to implement pharmacogenetics in psychiatry, using pharmacogenetics, in some cases pharmacokinetics as well, for two actions. One for all patients treated with the pot antipsychotics with aripiprazole and with risperidone, and also all the pediatric patients in hospitals. I mean, in hospital units treated with antipsychotic drugs or antidepressant drugs, they have pharmacogenetic profiles before they enter into the unit. Another important thing is cost efficiency. calculations is hard but necessary. I'm not going to talk about this, but I want to be brief on this. Also, it's extremely important the role of the scientific societies. In this sense, I think the Dutch guidelines and the Dutch pharmacogenetic working groups is a good example in Europe. In Spain, and Ana González is also doing a great work in this regard. Is the Spanish Society for Pharmacogenetics and Pharmacogenomics is trying also to produce common guidelines and to work on this. Also, it's extremely important a collaboration and organization at national and international level. In Spain, it's quite chaotic because we have 70 different systems of, the national health system has been transferred to the autonomous communities. So we have indeed 17 different health systems, and we need a common service portfolio and standard of cares. We need an implementation. Different models are possible. In Galicia, we have everything concentrated in a single unit. I think this is more efficient, but we need, but I mean that everything has to organize things in a different, in a way, in a possible way, but in the most efficient possible way. Education, I think, is extremely challenging and important. I love this review of Salgado in Nature some years ago, when they education is it's important for the clinicians. It's important for the patients to know that what is important to is possible to do, but also what is not possible to do nowadays. And I am pretty sure that if we are not working education, we can go to a treatment anarchy where off-label targeted drugs with limiting proof value are requested by patients or prescribed by clinicians, compromising the long-term potential of precision medicine to deliver benefits to patients and healthcare system. The future is obviously integration of biomarkers. Most of the biomarkers we have nowadays are genomic biomarkers, but in the near future, we still have some metabolomic biomarkers, little by little, proteomics, microbiome. But not only this, we have to include omics, but non-omic biomarkers. I have to do everything. We need to integrate biomarkers, but still we are far from integration. Have a lot of challenges. Probably AI, artificial intelligence, is going to have a role in this, but there is still a lot of challenges of integration. I have summarized some of them, but I don't have time to describe all of these. But lost values and class disequilibrium, noise and complexity of data, what we call the dimensionality paradox, more variables than samples, and obviously the heterogeneity and redundance of data. Problems that we have before the integration of biomarkers. And in this context, and also very briefly, Spain, through both the Ministry of Science and the Ministry of Health, through the Instituto de Salud Carlos III, they decided to bet for a national strategy on precision medicine. And as a first stage on this national strategy of precision medicine, they created they launched a program called Impact. I think this is an extremely ambitious project. I think the most ambitious project we ever had in Spain in the health system, in the Instituto de Salud Carlos III. Impact is a precision medicine infrastructure associated with science and technology. And it's the basis where we are now implementing all the national strategy with lots of programs and always thinking in translation and validation more than in research itself. Impact has three programs. One is a cohort of Spain. We didn't have any national cohort in Spain. Very far from the efforts we are doing in Germany, or imagine UK, but even Estonia. And I think we have a real need of a our cohort. This cohort is being coordinated by Marina Poyan from the Instituto de Salud Carlos III. The idea is a cohort of 200,000 people, representative of all Spain, with clinical data, image in some cases, and genomic data. And obviously also we need a data program. Imagine the amount of data that we are generating with different projects, and especially with this cohort program. So we have another program of an impact data program that is coordinated by Alfonso Valencia from the Barcelona Supercomputer Center. And the idea is to work in a scheme of federated EGA. EGA is the European Genome Archives. Now we have 2 nodes in Heidelberg and Barcelona, but in the future we are going to have in the near future some national nodes as well. And I'm coordinating the Genomic program. The Genomic program is the idea is a genomic infrastructure designed to be the basis of the implementation of all these genomic medicine in the national health system. And the idea was to is thinking in diagnosis. I mean, to perform diagnosis, but beyond the standard of care. Imagine, for instance, patients that they were negative for rare diseases after exome sequencing, they enter into the program, but always with a criteria of equality. The idea is to generate high-quality genomic data that, in addition to give that having a potential benefit for diagnosis, could be used in research as well. And we have decided to use 3 use cases. One is for rare diseases, another for cancer, and another for pharmacogenetics. We have some horizontal groups together with the other programs, with the data program, with the cohort program. We have been working a lot with ethical standards. Ethical standards are extremely important because, in addition to the clinical diagnosis informed consents, we have approved an informed consent for data sharing, very, very well designed with approval with legal standards. I have also an obsession with this, not because I am professor of legal medicine, but not because of this, but also obviously, but because I can see that some programs, as the Bavarian program in Germany, they have problems because they didn't have appropriate informed consents allowing for shareability of the data. And this is mandatory for inclusions of patients in our program. We have defined, we have set some standards for sequencing centers, for high capacity sequencing centers in Spain. We have in this moment 3. This is coordinated by CENAC in Barcelona. We have CENAC, Nacertic, that is a public company in Navarra, our center in Santiago. We expect to have in the near future another center in Madrid, and perhaps next year another one in Andalusia. And the three cases of use one was rare diseases and diagnose rare diseases in the country. For this, we have created a huge clinical network, impressive clinical network, where all the clinical centers are in there, with coordinators in each autonomous community, allowing that all the patients could enter into the program in equal terms, with equality. I don't have time to describe all the programs, but I think that it's very complex, the different committees we have. I think one important things are everything related with the HPOs, but also with the functional validation, because we are generating also a map of resources for functional validations of boost variants in the country. The inclusion criteria in this case is to have an exome sequencing completed. And we are going to use whole genome sequencing after that transcriptome, and HEPOs are mandatory for phenotyping. The second case, use of cases is cancer. One is for inherited cancer. More or less, this is coordinated by Gabi Capella at EICO. And the idea is more or less the same. All inherited cancer in the country, after, in this case, panels, negative for panels. And but we are also including in this use case cancer of unknown primary, because we know that these patients, CUP patients, could benefit from targeted therapies, and it's not in the portfolio of services of our health system. And this allows us to progress also in standards for somatic mutations. Standards for somatic mutation are extremely important, and this involves not only this involves even extraction, even quantification. For this, we have made a pilot project with 4 pathology services. We are taking advantages of a program where some of you are participating, that is the PICAS project, where we extracted here in Santiago more than 10,000 per samples, and we define also quality cutoff for sequencing. And the final project is pharmacogenetics and population genomics. The idea of this is to progress in accreditation, clinical guidelines for pharmacogenetics. We have some small pilot projects especially for ADRs for vaccines, but also we are working in defining standards for polygenic risk score for common diseases and trying to implement some pilot projects. Obviously, the main problem in this moment of polygenic risk scores for translation, for prevention, we have many challenges. The most important ones is to define standards, but also to validate prediction models and to improve imputation. In these regards, I think the most advanced genetic problem is obviously breast cancer. And this is because the huge effort that has been done in the world into the BCAC consortium. Imagine that the heritability of the breast cancer is not so high. It's only 28%, less than colorectal cancer. But we are far from the translation of, in colorectal cancer, for the translation of polygenic score to clinical settings. And but we are near to reach this with in breast cancer, and it's only because of that, of coordinated efforts at and we are going to do in Spain the first pilot projects for well, there are lots of challenges on this, but one important project problem for this is improving imputation. We are using in GWAS always. We are using always TopMed and the Michigan imputation, and if you see the results of PRS, they only really work well for the UK, but not for the rest of the countries. Population stratification has a very strong effect on imputation. That's why we are persuaded that we need to progress to make an effort, and we are doing this effort into the 1 million genome projects framework. In this, we have the idea of we have included a new working group that is the Genome of Europe. The idea is to have to have 500,000 individuals in Europe, representative for all the populations in Europe, sequence using genome sequencing. In Spain, we have decided to analyze it's 10% of the European population. That means that we have the obligation to sequence 50,000 individuals, and I hope we are able to get the budget to perform this, because I think this would be important. And that's all from my side. Just to thank all the people involved in this program. There are more than 1,000 people involved in the program. It's a huge effort that I think, and these people are not, I mean, they are only working together just because they think that this is good for improving medicine and for improving personalized medicine. And thank you so much. That's all from my side. I think I spent my 35 minutes, and that's all from. And thank you so much for the attention.
Okay, thank you, Angel, for the presentation. Let's move then from the we do the question. We have the discussion at the end. It's okay. So let's move to the second speaker. It's the Professor Paul Brenner. Let me introduce him very briefly. He's the head of the section of genetics in the International Agency for Research on Cancer. He got his degree in mathematics and medical statistics in the University of Leicester, and his PhD in genetics epidemiology. He joined in 1997 IARC as scientific in the Unit of Environmental Cancer Epidemiology. And in 2005, he headed the Genetic Epidemiology Group, and since 2009, he is the head of the section of genetics in IARC. In the last 20 years, he has been focused on developing international collaborative studies with the goal of identifying genetics and molecular risk factors for human genetics, with the goal to understand cause of cancer and enable early detection. He has been involved in many large international collaborative studies on head and neck cancers, lung cancer, and kidney cancer, and many of them also in diverse populations. He's leading the head and neck cancer working group within the EPI cohort, and involved in many analysis and publication focused on Mendelian randomization studies in cancer. As the result of his activity, has been published over 200 international scientific papers, and he got the Butker Medal for contribution to cancer epidemiology from the Trinity College in Dublin. And today he's going to talk us about the large-scale genomic studies on common and rare cancers to understand etiology and biology. So thanks, Dr. Brenner, for accepting the invitation, and please, the floor is yours.
Thanks, Anna, and thanks, Ankel, for the presentation. I mean, there's clearly a lot of points where, I mean, our research agendas overlap, and I think we can discuss some of those afterwards. So I'm going to give a very quick overview of the genomic epidemiology branch in the agency. And then I thought it would be helpful if we spent a little bit more time talking about two of the projects in detail. And that's the reason why Lynette and Sandra, they will take over the presentation to discuss those two specific projects. But first of all, I just want to say a few things about what the main goals of the branch are. So we're an international agency. So I think one of our goals is try to not replicate the work that's done in national agencies and at the national level. And our focus is a little bit more on some of the aspects you talked about, but trying to understand causes of cancer using genomic methods. I mean, I think as Anna pointed out, when I first came to the agency, I was in the environmental group trying to understand causes of cancer. And bit by bit, I saw that there was a great potential for genomics to inform us about cancer causation. And that's why my kind of journey has kind of moved more from the environment to genomics. There's also an awful lot of potential for us to identify biomarkers of or early detection of cancer using biomarkers, genomic biomarkers, but other blood-based or other forms of biomarkers. And that's a very important part of our work in the branch as well. So part of this can be studies looking at circulating tumor DNA or proteomic biomarkers. It's not something we're going to talk about in detail today, but it is an important part of the work of our group as well. And then we're also interested in trying to identify individuals at particular risk of aggressive disease, whether this is through germline work or looking at the somatic tumor as well. And finally, a lot of this work that we do helps us understand cancer pathways and biology. So just to mention the size of our branch, there's 14 scientific staff in the branch, supported by a similar number of technical and admin staff. We have a large group of young fellows, doctoral fellows, master's students who undertake an awful lot of the work with visiting scientists as well. Just to point out that we have 3 Spanish members of the branch. So there's Lynette, who will present today, who's leading a lot of the rare cancer work. We have Ida Ferrero, who is also who's mainly working on the mutograph study that Sandra will present. And we've just been joined by a fellow, Lorraine Torrance, who's joined us recently from Barcelona. So this is, an overview of the sort of works we do. I mean, we do still do a lot of traditional and molecular epidemiology. So we help initiate cohort studies in different parts of the world. We're active in the EPIC study, and then we undertake a lot of traditional genetic epidemiology. So this has moved on a little bit from the past when we were looking at high-risk families and the sort of work that David Golgar did at the agency, with a particular focus on breast cancer families. And this has more moved on to sorts of studies where we undertake these very large genome-wide association studies. So we lead these for several cancers. So we lead these large genome-wide studies for lung cancer, for head and neck cancer, for kidney cancer. We're intricately involved in the large GWAS studies for lymphomas as well. So this is an important part of our work as well. There's a lot of risk prediction and early detection work. So we do these ctDNA studies, proteomic studies, particular focus on things like lung cancer, on bladder cancer here. And also we are very interested in one of the topics that Angel mentioned on polygenic risk scores, looking at the utility of these, thinking of how these might be helpful by incorporating this information with screening studies. I mean, just looking at the data, I think one other cancer that I would highlight has been really interesting in this respect is prostate cancer as well, which I think perhaps even more than breast cancer is showing really interesting discrimination between individuals at high risk and low risk using polygenic risk scores. And then, of course, an awful lot of our work is based on the other genome of cancer, which is the tumor genome. And so this has become a very large part of our work. And the 2 presentations today will focus on these tumor-based studies. So I just highlight some of the ongoing collaborations that we have with Spanish research groups. There's this, this is yet to be announced, this first one. It's going to be announced in June, but we're leading one of these grand challenge projects funded by CRUK and NCI. This was a competition launched last year where they highlighted some questions with $25 million for successful projects. And we've been informed that we've been successful in getting one of these. And this project, which is looking at normal tissue and the genomics of normal tissue and the transition from normal tissue to tumor, is being led by us, but also by Nuria Lopez in the Institute of Research in Biomedicine in Barcelona, and by University of California in San Francisco. We have other. These are just some of the current collaborations that we have. We've always had a long history of collaborating with Spanish colleagues. At the moment, we have a collaboration with the Catalan Institute of Oncology in Barcelona on head and neck cancer as part of this Horizon 2020 project called Headspace. Another strong collaboration. This is a long collaboration that we've had on lymphomas that goes back over 25 years. And a large part of this is on the genomics of lymphomas and coordinating large genome-wide studies. We have a very interesting collaboration that's developing with Luis Montuenga from the University of Navarra on biomarkers for early detection and prognosis in lung cancer, with a particular interest on using proteomic arrays. And finally, we have this collaboration on rare cancer genomics that Lynette will talk further about. So the 2 specific examples, just to give some flavor of the work we're doing, that we're going to spend more time on, is the mutograph study. This is a previous grand challenge study that was funded in 2017 that is still underway. And Sandra will talk about this, and then Lynette will talk about the Rare Cancer Genomics Initiative. So perhaps at this point, I can pass over to Sandra, who will give some more details on the Mutograph study. So
thank you, Paul. I'm going to share my screen now. Well, before I start, I will introduce myself. My name is Sandra Perdomo. I'm a scientist working in the Genomic Epidemiology branch. And I have to say that it's a pleasure for me to be discussing today with colleagues from Carlos III. I'm going to tell you a little bit of an anecdote, a personal anecdote. When I finished my PhD, I had the opportunity to work with a group of Marina Poyan that Angel just mentioned in his presentation during 6 months in the National Epidemiology Center. And it was back then when I started kind of my first introduction to cancer epidemiology. So it's always nice to have the opportunity to go back and discuss with those people and that institution that had somehow an impact in your career. So it's a second motivation that I have to be here today. So I'm going to present you today 2 examples of genomic epidemiology studies that we have in our branch in the area of cancer prevention. The first one is the Mutographs project that is aiming to identify novel causes using the analysis of mutational signatures. And the second one is the most recent initiative that Paul just mentioned, and is trying to understand cancer promotion using the study of mutations in normal tissue, and it's called Prominent. So the Mutagraphs project is a CRUK Grand Challenge project that is aiming to understand the causes of cancer through the studies of mutational signatures. This is a collaboration that we have with the Wellcome Sanger Institute, led by Mike Stratton. Paul is leading the team here at IARC, and we also have the collaboration of very recognized scientists in the University of California, San Diego, in San Francisco, in King's College. And this project is based on the concept that environmental exposures and also endogenous processes, cellular processes, can leave a trace in our genome. These are specific mutational patterns, and this is the definition of a mutational signature that can serve as a mutational fingerprint that can be linked to a specific cause of cancer. So using this concept of mutational signatures, through the Mutagraphs project, we wanted to identify mutational signatures in 5 different cancer types, including 5,000 cases across 5 continents. These 5 cancer types are colorectal cancer, kidney cancer, pancreatic cancer, esophageal squamous cell carcinoma, and gastroesophageal adenocarcinoma. Why we chose these specific 5 cancer types? It's because we see that these cancer types have a different incidence across different regions. And actually, the risk factors that we know for these 5 cancer types cannot fully explain why we see these differences in incidence. So we think that using mutational signatures, we might identify additional causes that could probably explain better why we see these differences in incidence for these 5 cancer types. So in this map here, I'm showing the number of cases that we have collected all over the world in different countries and for the different cancer types. We have a very standardized sample collection protocol, but we have also a very extensive lifestyle questionnaire with a lot of additional clinical data for us to be able to associate different exposures with the mutational signatures. We also had the opportunity to extend this project to 3 additional cancer types. These are and based on specific exposures that we wanted to explore or a specific distribution in different countries. This is the case of head and neck cancer, where we want to kind of better describe a mutational signature that could be associated with alcohol consumption. Urothelial cancer, and we're really interested in seeing the possible link between the consumption of opium and the development of this cancer in Iran. And gallbladder, that is a rare type of cancer that is very prominent in India, and especially in the north region of India. To give you an idea of the numbers and the amount of samples that we have collected and that we are processing right now, we have received above around 7,000 samples all over the world here at IARC, and we have processed at this point 75% of these cases. We have a very detailed and very stringent histopathological review. And right now we have released over 2,000 sequencing, whole genome sequencing from tumor and germlines. Our first priority for analysis was the analysis of esophageal squamous cell carcinoma. And this is the work that was led by Sarah in the Sanger Institute and Sergey here at IARC. And we published the analysis last year in Nature Genetics. And actually one of the main results of this first publication was that the mutational profiles of esophageal squamous cell carcinoma are extremely similar across countries. And actually it was the ApoVec signature the one that we found in the majority of these cases. I will go back to this result because this is actually very important for the launch of the second project that I'm going to mention. The second scientific priority, and this is actually the work that we're doing right now, is kidney cancer. And we're focusing on whole genome sequencing data from above 900 clear cell renal cell carcinoma cases that were collected in 11 countries from different incidence rates. And we're focused on our analysis in these countries and also in known risk factors like hypertension, high BMI, tobacco consumption, and family history. You see here in the table the number of cases that we have collected distributed by different countries and by different incidence regions. And I'm going to show you very, very preliminary results of this analysis. I've identified 11 signatures that were extracted. Most of these signatures are made up of known COSMIC reference signatures. COSMIC is the largest catalog of curated signatures that we have right now. And I'm just going to mention two of these results from the 11 signatures. One is the signature that is associated to Isolocic acid exposure. And then the signatures that compose signature 4 that doesn't have an etiology assigned yet. So this Isolocic acid signature is Isolocic acid is a risk factor that has been already associated to upper urothelial cancers, including kidney cancer in the area of Romania. But in our analysis, we have identified an additional signature that is also associated with the Isolocic acid exposure. And here we're seeing for the first time, we're not only seeing these signatures in all the Romanian cases, but we're also seeing in the majority of the Serbian cases at a lower levels, but it's the first time that we identified it in Serbian cases. So this actually is suggesting that probably there is a wide population exposure to Isolocic acid, much higher than what we probably previously assumed. And right now, well, we are performing some geospatial analysis to try to shed more light into this exposure. The second preliminary result is this component of that signature 40, that we see that it strongly associates with the incidence rate of kidney cancer. For the moment, we haven't seen any specific link with other risk factors that we know for kidney cancer, but this could be actually an indication that there is an unknown exposure that actually is responsible for this variation and incidence. And this is actually one of the objectives that we have in the MutuGraphs project. So we are kind of performing additional analysis to try to get some more information on what this signature means and what is driving these differences in incidence in kidney cancer cases. The next step for MutuGraphs will be to complete the analysis for the other cancer types. So the next one is head and neck. We have completed the sequencing, whole genome sequencing from 265 cases. Our analysis will be focused on alcohol exposure, HPV, and the microbiome. Colorectal cancer, we have completed 1,000 cases of colorectal cancer from different regions that are just listed here. And the sequencing is undergoing right now. gastroesophageal junction is the next one, and we'll be focus our analysis in high BMI and exploration a little bit of the microbiome, especially Helicobacter pylori infection and reflux, and pancreatic cancer, in which we will use this analysis to try to understand better mutational signatures in young onset of pancreatic cancer and in cases with a strong family history and also a history of diabetes. Now I'm going to move from mutagraphs, and I'm going to very briefly present you the new initiative that is called Prominent, Discovering the Molecular Signatures of Cancer Promotion to Inform Prevention. Actually, I think this is the first time that we are presenting this project outside IARC. So you have you are the first ones in hearing about this project. And as Paul mentioned before, this is a project that is called by Nuria Lopez Vigas in the Institute for Research in Biomedicine in Barcelona. Paul is leading the team here at IARC, and Alan Ballman is leading the team in the University of California, San Francisco. And we have also collaboration with other co-investigators also from here, from IARC, Mark Gunter, and other collaborators in the US and in Sweden. So now in Prominent, we are no longer interested in looking at the mutation profile of tumors, but now we're switched and we want to understand the mutational profile on normal tissue. And this is based on I think it's in the last decades that we have evidence that normal tissue cells can accumulate a high frequency or known driver mutation. And this is being published in different histology types. And this is there is a strong evidence that this occurs. There is also a second part of evidence that that drives this new project, and is one of the results that I previously mentioned for the esophageal cancer paper, and is that many of the known risk factors of possible carcinogens are not really mutagens, and this is also seen in many of the studies conducted in mice. So many of these possible risk factors do not act as mutagens, but somehow we think that they can promote the development of cancer. And this is actually the hypothesis that we want to test in Prominent, that promoters can stimulate the growth of these cells in normal tissue that carry specific driver mutations. So, well, the main questions that we want to respond in this project is, first of all, what are these factors that promote selection of these normal cells with specific mutations? Are the same high risk factors for the different cancer types that we thought were mutagens, but no, they have a different mechanism of driving cancer, and probably is promotion and not mutagenesis? What and where are the cells that harbor these mutations? So that's why we're going to test different cancer types, and we're trying to locate what are the specific cells that actually contain this mutation and can drive the development of cancer, and what are the mechanisms that promote the neoplastic growth. And in order to respond to these questions, we have gathered a kind of a very multidisciplinary team, and we want to kind of approach this question from different areas. And that's why we're going to be using a lot of human samples. Actually, we're going to be using a lot of the biorepository that we have collected as part of Nutrigraphs. We'll be looking at the normal tissue of cancer cases, but also normal tissue of non-cancer patients, and cases from, again, low and high risk incidence regions. We will be also working with mouse models and organoids to try to understand how these promoters can select specific cells and mutations in different tissues. We are going to use state-of-the-art technologies and analysis like spatial profiling, in situ mutation detection, a very, very sensitive single molecule sequencing to be able to detect the mutation in the normal tissue. We're going to be doing some CRIS screens to try to identify the genes that can regulate these tumor promoter phenotypes, and And we also obviously will going to include a lot of the bioinformatic analysis, very specific bioinformatic analysis to try to model the molecular evolution of the normal tissue into the neoplastic transformation. So at the end, the key delivery of this project will be to be able to understand promotion, but also to try to identify if these promotion events are preventable. So if there is a promotion event that is preventable, and obviously this will have an important implication for public health, but if not, we can also identify possible agents that could reverse promotion. And this is also another deliverable of this project, a little bit of the concept of chemoprevention to try to see if that promotion step can be reversed. And I think I'm gonna just stop my presentation just by showing you, I'm here representing a large group of people who are kind of work together with these projects. And this are the faces of some of the people that contribute to this project from here from IARC, but also there are a large of people contributing to the project from the many collaborative centers that are part of Nutrograph and that will be part now from of Prominent. And with that, I'm going to finish. I'm going to stop sharing and I'll give the floor to my colleague, Lynette.
Thank you for this opportunity to present this data. And first of all, just a brief introduction of my background. So I studied in the University of Oviedo. I studied biochemistry there. And I must say that, well, now with all the things that you Spanish scientists know about, Carlos López Otín was a mentor and a person who in fact helped me a lot in my career. And then I moved to IARC for a PhD under the supervision of Pierre Renaud. And I work a lot on breast cancer there. And then I moved to the Max Planck Institute in Germany in the group of Roman Thomas, who was leading the research on cancer genomics on lung cancer. And I think maybe the highlights that I can give about my background was the discovery of NRG1 fusion genes in lung cancer, specifically in adenocarcinomas. And also pioneering the largest genomic studies on lung neuroendocrine tumors, including small cell lung cancer, large cell neuroendocrine carcinomas, and pulmonary carcinomas. Then I joined IARC. I was back to IARC as a scientist in the genetic cancer susceptibility group led by James McKay, where I had the opportunity to start the Rare Cancer Genomics Initiative together with another scientist in the same group, Matthew Fall, where we have tried to expand a little bit our cancer genomic studies to other rare cancers. And this is what I'm going to talk to you today, giving you a snapshot of what we are doing within this initiative that further this year just translated in creating the Rare Cancer Genomics team within the new organization at IARC. And I just put a couple of things to link a little bit with the presentation of Angel, which I found it was really interesting and definitely accurate in describing the limitations that we have and the difficulties that we have now with precision medicine. So with this, just a brief overview, what are the rare cancers? So they are those that have an incidence below 6 out of 100,000 people per year. And although they are considered rare, collectively, they are quite common. In fact, they represent 25% of all cancer diagnoses. And because there is this lack of basic biological and clinical knowledge, due to the challenges that are inherent to the rarity, they have usually a worse outcome than what we usually see in common cancers. And I think this is important to make the first point to what Angel was saying. We are considering the rare cancers, but with these molecular studies, the rare molecular subtypes of more common cancers have the same challenges that we face with rare cancers. So this is something that is important to keep in mind. So what is this initiative? This is an international multidisciplinary effort aiming at shedding light on the molecular characteristic of rare cancers with the major goal of understanding the etiology, carcinogenesis processes, and of course, ultimately improve the clinical management and the prognosis of the patients. So within the 12 family of rare cancers that have been described by the Europe Rare Cancers Project, we are focused on 3 of them. One is the interest we have on studying malignant pleural mesothelioma, which is a disease caused by the exposure to asbestos. We are very interested in neuroendocrine tumors, most particularly in aggressive lung neuroendocrine tumors, but also in the general progression from low-grade to high-grade neuroendocrine neoplasms. And more recently, we have started a project focused on soft tissue sarcomas and carcinosarcomas. So for all of these studies, we team up with very well-established biobanks that provide high-quality material for deep genomic analysis with centralized pathological review and with detailed clinical and epidemiological annotations. So what is our research approach? We initially focus on a multi-omic bulk sequencing, more particularly integrating methylome, transcriptome, and genome analysis for our initial analysis. And now we are expanding this to include multi-region, single-cell sequencing, and also spatial profiling. And more recently, we have started working on artificial intelligence on whole image scan, on whole scan images. So this data, to get the maximum from the data that we generate, because this basically the description of an integration of all an interpretation of this data is led by Matthew and myself, but we team up with Nicolas Alcalá, who is another scientist in the team with a strong background on cancer evolution. And then we also have a very strong collaboration with Talia Dayton, who is currently a postdoc in the Hans Clever group, but now was recently appointed as group leader in EMBOL Barcelona for her work on organoids, which she's going to pursue. And we will keep working together with her to have more mechanistic insights on our molecular findings. So I'm going to give you just a snapshot of the scientific highlights of our projects, but if you're interested in what we are doing, I invite you to visit maybe our website for the Rare Cancer Genomics Initiative and for the Rare Cancer Genomics team. So for the Mesomic project, and again, going a little bit what Angel was saying, we try to focus here on the unknowns in patients stratification. So this is a figure of our recent paper, which is under second round of revision in Nature Genetics, but you can have access to an earlier version in bioRxiv, in which we have analyzed more than 100 malignant pleural mesothelioma samples, in which we have generated the first and largest whole genome sequencing data series that we have integrated with transcriptome and methylome data. And something that we have wanted to do was this, and basically for the input from Nicola, was this unsupervised analysis, where we are not taking the histological classification that divides malignant pleural mesothelioma in 3 groups and try to further define this classification. We have let the data talk to us, and we have performed multi-omics factor analysis, in which we have identified 4 independent sources of molecular variation. You can see here the ploidy factor, the morphology factor, the adaptive response factor, and the SIM factor. And interestingly, when we have correlated our 4 latent factors or factors with the current pathology and molecular and digital pathology artificial intelligence Paris score, we have seen that all of them correlated very strongly with our morphology factor, but only with our morphology factor, which means that the other 3 factors have been previously disregarded. And this is particularly important. I always like to compare malignant pleural mesothelioma with another recalcitrant cancer that you may be very aware of, small cell lung cancer. Small cell lung cancer has frequently seen as a very homogeneous disease, has been treated as one disease, and immunotherapies has been strongly disappointed in these patients. But more recent stratification based on expression patterns have shown that, in fact, there is a group of small cell lung cancer patients that may respond very well to immunotherapies. So this is very important, but if we are not aware of the sources of variation, we cannot take them into account when we are thinking about patient stratification. And this is what we think our study is very important for. So our ongoing work now is further characterizing how the intratumor heterogeneity of malignant pleural mesothelioma impacts these molecular characteristics. We do this through multi-omic, multi-region single-cell sequencing, and also further characterize the tumor microenvironment interaction that seems so important in this disease through spatial profiling. In the case of the lung genomics project, I think the discovery that we want to highlight is the identification of supracarcinomas. In fact, we have done also multi-omic analysis. Here, each dot is one sample, and we have, again, through unsupervised analysis, study a little bit the characteristic of the low-grade, low-aggressive pulmonary carcinoids and high-grade, high-aggressive large neuroendocrine carcinomas. And very interestingly, we have identified 6 carcinoids that have the morphology that is expected for a low-grade, low-aggressive tumor, but the clinical and the molecular characteristics of a high-aggressive, high grade. And this is important because one of the major issues in pulmonary carcinoids is that there is a subgroup that will relapse, that will have an aggressive disease, metastasize and relapse. And right now, all the patients need to be followed because we cannot identify these ones from the beginning. So when we identify these tumors, this has led us to have the opportunity to participate as co-authors in the recent classification of thoracic tumors, to include this question: should grade 3 lung neuroendocrine tumors be included in the classification of thoracic tumors, in the same way that they are already included in the neuroendocrine tumors classification of the gastrointestinal tract. And now, of course, we need further evidence because we only had 6 samples, but we have now increased the number of samples of these aggressive diseases, and we are now performing a comprehensive molecular characterization of those. Another thing that was important from this study is that we were able to identify within these molecular groups particular expression patterns that may help us to identify these patients that may need follow-up. And this is a study that we are doing now in collaboration with the European Neuroendocrine Tumor Society Task Force to see if these genes can be in fact implemented in and included in molecular imaging procedures to identify these aggressive pulmonary carcinomas. So before going to the last highlight that I want to mention, I want to stop here to discuss a little bit about the useful open science that we are so committed to. And that I think, again, Angel, you mentioned these limitations that are definitely important also for us. We really want to go beyond what is a raw spreadsheet PDF model. And we want to provide the full spectrum of open science data and tools to maximize the reuse potential. And this is particularly important. I think this is important for all genomic studies, for all studies in general, but particularly important for rare cancers because sometimes due to the limited amount of samples, we need to pull data from different studies, and it's very difficult and time consuming. So we really want to facilitate this task because we have been through this and it has been painful. So first of all, our data is always in the bioarchives from the moment the paper is published. Then we usually publish a data note, which is associated to our main paper, in which we describe the quality controls on all the analysis that had been done. So people do not need to go through all of this. They already know this, and they know which kind of data they are facing to. And then we also provide open codes because we want to, of course, one of the major issues today in science, and it's very important, is the reproducibility. So we want our results to be able to be reproduced. And then finally, open resources, what we call the interactive tumor maps, well, has been called interactive tumor maps. And I think this is particularly interesting for pathologists, clinicians, and all those who do not have a strong background on bioinformatics. So what you can see here in this video is the way you can use these interactive tumor maps that are open for everyone. You can just choose your favorite gene and just try to check what is the different expression of this gene in the different groups that we have identified. And this is particularly interesting also for hypothesis testing. Another way to use these tumor maps, how this can help the study of rare cancers, is the last highlight that I want to make on the Pangenomics project, this work that we have with Talia. So Talia has generated this biobank on patient-derived tumor organoids for neuroendocrine tumors. So Nicola has led the analysis of the multi-omic sequencing data that was generated. So he used these tumor maps to project in these maps the organoid and the primary tumor. And this has helped us to see that, in fact, these PDTOs conserve the global molecular profile, the intra-tumor heterogeneity, and the mutational processes of the original tumor. So now we are in the good shape to use this established NEN biobank to model the disease initiation, and particularly this progression from low-grade to high-grade neuroendocrine tumors. So then just my last slide is just to give you a snapshot of what we are doing in the Sarcomics project, particularly because I've seen that you have some groups with interest in these tumors. So here, what we want to do is really understanding these sarcomagenesis processes. We consider that it might be an option that this when we focus on soft tissue sarcomas with complex genotypes, they can go from well-differentiated to extremely poorly differentiated, like the undifferentiated pleomorphic sarcomas. And all of them have, at the morphology from the morphological point of view, they have mixed patterns. So this progression might indeed be possible. But not only for soft tissue sarcomas, also for carcinosarcomas, we think that the processes might be the same. So even if now they are classified as different groups and studied as different groups, if we are able to prove these common processes, we might be able also to identify common therapeutic vulnerabilities against these aggressive tumors. So this is where we are focusing right now on the Sarcomics project. So as a name for this presentation was trying to identify how we could collaborate. I have look at the groups that are working on this. I identify a couple of groups that are working and interested in rare cancer. So I would be very happy to discuss with you further if you think this is interesting for you, and particularly also for the work package 2 of your Impact Genomics project from Angel. So in the meanwhile, I would like just to thank you all of you, and I will be happy to answer your questions. Thank you very much.
Okay, thank you, Lynette. Thank you for all of you. So I think we have, Pilar, some time to have.
I mean, we can continue with the discussion unless one of the speakers are really in a rush. We can dedicate 15, 20 minutes to discuss if you're okay.
Yeah, I mean, also people can also write the questions if you want in the chat, and we can read the questions to the speakers. All right. So let's. I mean, waiting for questions. Maybe I can make a general question for the speakers, and it's been. It's been related with the PRS and to estimate population risks for most of the common diseases, as you have mentioned in your talks, or Angel also mentioned that there is a thousand. It's going to be 1,000 of hundreds of PRS to estimate population risks in a short time. So having this in mind, and in your opinion, which will be the best strategy for the next decade to implement successful these PRS and these risk models in the clinical practice to improve the disease prevention and population health? It's a very complicated question, but I mean, just to see your view about this. How do you think we can do it in order to to be success with this?
Maybe, Anna, I can give an immediate response to that. There was a lot of discussion 2 weeks ago at the AACR, the American Association for Cancer Research, their conference in New Orleans, about PRS and how they're evolving and the potential utility and the opportunities and the dangers of PRS. And I think it is clear that for certain cancers, for common cancers, there are one is able to distinguish high-risk groups and low-risk groups. And, the data is emerging quite comprehensively for prostate, for breast, and even some very interesting data on colorectal, I'd say. That, when you apply these, and some of this has been published recently from some colleagues in our group as well, applying the latest scores to UK Biobank, you can show, really quite strong distinction. And the question now is that, well, there's a couple of issues. One is, how interpretable these polygenic risk scores are in other populations. And
that's
I think Angel pointed out, the further you get away from, the Western European kind of population on which most of this data is based, the less accurate they become. And so there's this need to somehow, develop these in broader populations. And I think the other one is what's the potential utility of this? And perhaps the potential utility that's emerging is somehow to incorporate into ongoing cancer prevention and screening efforts where these are working. So clearly for breast cancer, can one use this information to decide, well, certain women, maybe they should be screened earlier, more frequently. Certain women, maybe later or not at all.
That's right.
And similar questions for, for prostate cancer, for certain men, perhaps their risk of prostate cancer, if they're in the bottom quartile of genetic risk, maybe they never, they don't need to undergo particular screening strategies. And similarly for colorectal cancer. And, and I guess this is where the problem comes in, because it's a question of implementation and how this information can be implemented. I mean, it's not available at the moment, but if it was available on a population level, how this information could be implemented into screening programs without damaging those screening programs, and in a fashion that was interpretable and useful for individuals, for the population as well, where individuals might accept more intensive screening or might accept less intensive screening. So there is there's a couple of barriers. There's a barrier of not having this information on the population level. And the second is how it can be implemented in a way that enhances ongoing screening programs. So I think that was a little bit of the conclusion of some of the discussions that were emerging at the AACR a couple of weeks ago. And I tend to agree with that kind of situation at the moment.
Well, I can add something to this. Well, first, there is a classical British joke that concerning, well, talking about translation of this to practical terms, to real clinical practice. It's how porcupines makes love. It's very carefully. In my opinion, we should do that also very, very carefully. First, because we need still to work in data and standards. I know that all these direct to consumer genetic testing, they are offering things like that. But for me, this is very wrong. And at least I could try not to apply this to our health system. So I think we need to progress in small pilot projects where the different projects that we have for and different standards that we have implement can be solved. That's at least my idea here, because it's not only that we need we need data to see how this type of models work in a given population. We need also to agree in common standards. We need also to agree in how to implement all the clinical information and how that lots of different things. And obviously, as Paul mentioned, without entering into conflict and coordinating this with the screening programs. I think we can do this for breast cancer. And my idea is to do that because we can learn also some other things that we have for instance, ethical issues. For instance, how the people can react on this. I mean, there are social and ethical issues also involved on this as well. So I think we need to very, very carefully to try to implement this little by little and to always with scientific standards and scientific evidence. And that's my opinion. I think that we should start with these pilot projects and I can see that this is very near in breast cancer because we have more or less the SNPs we need to use. We know the information we need to add. We need or even mammography density. Can we add this? We have body share and we have prediction models quite well validated. So I think that's a good example to start with, I think.
Mm-hmm. All right, thank you. I don't know if there's any other comment. I mean, I have other, I can continue asking if there is no questions from the public. So maybe I would like to ask Paul Brenner. It's been related with the cancer early detection, which seems to be the best strategy to decrease the mortality, of course. And you have been talking, you have experience in biomarkers and in liquid biopsy, let's circulating tumor cells, proteins, epigenetics, circulating tumor cells. But in your experience, which is the most promising type of biomarkers in terms of prediction performance? People are working on CTCs, people are working in circulating tumor DNA. Maybe it's a kind of combination of many of them, or you will focus your effort in a particular type of biomarkers or predictive biomarkers in liquid biopsy. What do you think?
This is clearly an incredibly challenging field.
Yeah, it is.
You know, and perhaps we were a little bit naive, you know, kind of 5 years ago or before, and thinking that this would be simpler than it's turning out to be. I mean, I think for some of the very large initiatives and the private initiatives, those funded by Illumina, etc., the GRAIL initiative, that is, you know, investing an awful lot in ctDNA. I think even they are finding this to be more challenging than perhaps they expected it to be. And it's unclear if a lot of this is going to work. I mean, it might be just the nature of kind of early cancer detection that, that there is not enough circulating tumor DNA that's identifiable at an early stage when it's a treatable stage. And there is even emerging evidence that for some of the very early tumors that are detected for some cancer types, very many of them already contain, kind of subclones that contain a mutation that results in metastases. So it is becoming incredibly challenging. But we'll see how some of these large initiatives turn out. I mean, I think it will depend on the cancer site, and there will be different technologies that are more kind of more reproducible for specific cancer sites. In our group now, there's a very active program on bladder cancer that is showing promising results for circulating tumor DNA that's focused on urine available ctDNA for the TERT mutation that is showing really interesting results. I think our most promising one as well is for HPV-related antibodies for head and neck cancer, which again is showing incredibly promising results, but it's a very rare cancer. So, on a population level, one would not undertake population screening at the moment based on that, but one can detect these antibodies many years prior to diagnosis. The one we're investing the most in at the moment is in some of the proteomics work.
Genomics.
And using kind of these various Olink type panels to identify different combinations of proteins available in blood samples that are predictive of early lung cancer. And our initial results, and with the way we think this is going, is that this will help us to identify individuals that need to undergo screening. So it will provide provide a more accurate idea of the subgroup of individuals at high risk who should undergo low-dose CT screening. So that's where most of our work is at the moment. I think the final thing you mentioned, Anna, and what most people probably realize is that it will take a combination of these different technologies, and that maybe together, we will be able to have the most optimal approach. But it is incredibly challenging.
Yeah, I suppose. But it's been done. Sorry, it's the studies are done in people already with the disease, or it's healthy people to identify the biomarkers.
So our work is in healthy people, and this is one area where the agency has kind of a long history. Well, first of all, with the EPIC cohort.
Yeah. Okay.
Then in bringing many cohorts together. So we work with large cohort consortia where we bring a dozen or more cohorts together to because clearly one wants to look at those blood samples or those urine samples that are taken the 2, 3, 5 years before the diagnosis occurs. So our strategy is often to develop these initially in case series in individuals who have the cancer, and then test them in the cohort samples.
Mm-hmm. Fine. All right. So, Pilar, we can hear you.
Yes, I'm gonna allow James McKay, who was saying something about the previous comment. I don't know if he's there.
Hi. It's
hi.
Great to talk to you all. It was just a quick comment about the comment of earlier about the genome-wide association studies, and I fully agree that there's a huge potential that we have, but the only thing we shouldn't forget is that at the moment, the 3 that are working so best are also the ones we've studied the most extensively: lung, prostate, and colorectal. And we don't want to put aside the other types of cancer that could be equally applicable. And so one of the things that we need to work in, particularly at the agency, is to try and expand the sample size of some rare forms of cancer: lung cancer, lymphomas, kidney, head and neck. And that's a large initiative within the branch as well. And I also, one of the biggest challenges is going to be for those types of things. You rightly pointed out that the difficulty of translating these polygenic risk scores across different populations, and that's going to become even more acute when we start looking at rarer cancers, because these samples are going to be even more rarer. So I personally think the agency has a very big role in coordinating those types of studies, putting these samples together to get us to the sample size we needed. Thanks for the opportunity to have a chat.
I agree. Okay, so I don't know if Pilar, I have another question. If you want, I can continue to ask questions.
There is no
but
more
I
questions from the audience. If maybe the speakers want to
Yeah.
ask each other also.
Oh, that's great.
Be
That's great.
completely free.
Sure.
Well, I guess I was very interested to hear about the impact cohort that's been developed in Spain of 200,000 people. And I guess one of the questions I had was, does that have a main focus? I mean, will it be large scale genotyping or sequencing of that cohort, as in UK Biobank, or what is there one particular focus, or are there many different objectives for that cohort?
No, it's very similar to the UK Biobank cohort. We are going to have genotyping data of all the 200,000 samples and whole sequencing genome data of 50,000 samples. I think in this case, perhaps the clinical information is more complete than the UK Biobank, because and it's also with follow-up of the patients. But I can see too much differences, I mean, of the two initiatives.
Okay.
All right, we have a question from the audience. It's I can read it if you want. It's Elena Urcelay. She's asking to the speakers. She said, or she commented, it's a bit frustrating that after the GWAS performed in every possible trait, and after the integration for epigenomic studies in complex diseases, not even a gross subclassification of patients is available according to molecular pathways affecting subgroups of patients. Is the lack of clinical information of patients the main bottleneck, or enough regulatory data is missing?
I think from the cancer point of view, most of these studies, they rely on cases that do not have a detailed kind of genomic classification of the tumor. There is often pathology information available, so there will be some histology information available, and that's often incredibly useful. So for example, if you look at the lung cancer genome-wide studies, there is an awful lot of, there are enormous differences in the results that one finds when one considers the results by histology, and the analysis has to be done by stratifying by histology. But going beyond that, it's not, well, the information is limited. As I said, there's usually not complete genome data on even a subgroup of the cases. So that's very limited. And that's one major reason why it doesn't go down to greater detail.
Well, I could add something to this. Obviously, to stratify disease with the data we have is always difficult, obviously. But in some cases, I think not only GWAS, but large-scale sequencing studies is allowing nowadays to stratify disease. In one good example is autism, spectrum disorders. Obviously, it's very genetics in the sense that the heritability is so high, and 50% is functional rare variation, and the rest is common variation. But now we know that more than 150 genes are involved in autism, but not genes random in the genome. They are concentrated in three main groups representing more than 80% of the cases. One are genes related with synapses, other group of genes related with neurodevelopment, and other group of genes related with histone modeling. So that's a way to stratify. And little by little, I think we are being able to stratify. But obviously, from SNP to the causal gene to the pathway, still a lot of work to do. But I think we are little by little in the right way. I don't feel frustrated for the results of the GWAS. Perhaps the people have the expectations about that, but I think they have given an extremely good contribution to the understanding of the disease and to the certification of disease in many cases.
Yeah. I mean, maybe also it's maybe something maybe we have to, I was just a comment is maybe, do you think that also we have to put the focus on the non-coding genome that is not being explored very well till now, or maybe past because we haven't done genome-wide association studies, we are doing now case control exome sequencing, and do you think the whole genome in case control studies will be the next step for cancer susceptibility? Or we don't need it?
Well, I think we'll find an awful lot, Anna, from the sequencing of UK Biobank. This will give us a strong indication of whether kind of rare variants are, are important in explaining a lot of this unknown heritability that the GWAS studies have not identified. So I would kind of wait a little bit and wait for that analysis on the whole genome sequencing that is emerging from UK Biobank.
Yeah,
what that tells us, and
we'll
if
see.
that's very promising, then we might need to go back to a lot of the cases, a lot of the DNAs in our biorepositories to undertake sequencing there.
That's right. Yeah. Okay. So
There is no more questions, Anna.
Yes, sure.
No. There is no more questions from the audience. It's just that I don't know if you want to. You are all very busy, and I don't want to keep a long time.
Yeah. I mean, I was just would like if I have time to have a question to Sandra, because I thought I see that the Mutograph project is very interesting. Just a curiosity, it's about the microbiome data that you plan to integrate it. Is that right?
Yeah, for the different cancer types that we're analyzing and that are part of the Mutograph project, we want to include the microbiome analysis. I mean, this is the case for head and neck, for example, which is there is a new evidence that the microbiome, obviously, we have HPV related tumors there, but also the composition of the microbiome might have an impact, for example, in the survival of head and neck cancer cases. And we want to include that type of analysis using the whole genome sequencing of the cases that we're doing as part of Mutograph. But we also know that the microbiome is also important and is emerging as an important etiology, or the composition of the microbiome for other cancer types is the case, the gastroesophageal adenocarcinoma is the case of colorectal cancer.
Yeah.
So we want to incorporate this analysis as part of the analysis that we have planned in Mutographs.
Yeah, gut biopsy, from gut biopsy as blood sample, or
yeah, I didn't go to the details of how we process the samples, to be honest, but the majority, especially for the gastroesophageal adenocarcinoma cases, majority of the cases are coming from biopsies. And as I said, we have a very detailed and stringent histopathology review process. And many of these cases now have to go through a laser capture microdissection to be able to kind of capture a good amount of tissue with enough percentage of tumor cells to be able to sequence a large amount of tumor cells to be able to get good sequencing information from these cases.
Okay, great. Really, really interesting. Yeah. Thank you.
There is Elena Urselay who was asking the previous question. She's saying that she's also a believer. Of course, she was just trying to make a provocative question. And she just congratulations the speakers. And then we have another comment from Antonio de la Vieja. He said that maybe perhaps we have to make an adequate classification. It will be necessary to wait for all the omics, environmental factors, and microbiotics to be completed, but perhaps we will arrive at an absolutely individualized medicine. Maybe that's kind of questions for both the speakers, and then maybe we can close up. Do you think we will get to a completely individualized medicine?
I think we need many of the new biomarkers from the fields. They always need validation, foundational validation, and validation in the application. Then I was also talking about the challenges we have in the integration of the information. That is not so easy. but obviously it's a long way, but it's something that we need to do little by little. That's all from my side.
Do you want to add something, Paul?
Well, maybe, we as we clearly understand now, every cancer is unique. So in that sense, that is driving us down to this concept of individualized medicine. But then I think there are dangers as well with this approach. And I'm kind of a little bit skeptical about some of the successes in individualized medicine that have been made within cancer at the moment. And I think the approach is going to be limited, and we need to kind of still keep the idea of population level kind of interventions. I don't know if Lynette or Sandra want to add anything.
Yeah, well, I think we are somehow biased here at IARC because we always think about population, public health, and population-based studies. So that's why, and it's going to be difficult to find exactly that balance into what is individual and personalized medicine and therapies that could be useful for many of the patients, but also those public health interventions that could probably prevent many cancers, but can also could be useful for detection, for early detection. So I think the right answer would be to have a balance between the two of them, but also and relying to what Angel said before, with evidence and with replication, with a strong evidence to be able to kind of implement these new technologies. This is the most important thing.
So,
okay, then maybe we can close up unless somebody wants to say something at the end. I just want to thank all of you. This is an idea that we got to get more connections between the Instituto de Salud Carlos III and actually not only from ISCIII, but also from other groups working in Spain and IARC, letting know what IARC is doing, but also letting IARC know what some initiatives in Spain. And for that, I'm very thankful to Angel because he presented the impact project, which is also a big idea of the Institute. And I'm happy that now you know it and maybe you can connect with Angel and with Marina and make this more approachable. And any connection that comes from this, it will come by itself. So thank you all for presenting the data so that the people have ideas for collaboration. And I just want to finish. Well, I thank Anna for the effort here being chairman today. And I want to just announce the next seminar, which is supposed to happen in June the 3rd. And it's going to connect, the speakers will be Freddy Bray and Maria Jose Sanchez Cuellos. And they're going to talk about cancer surveillance and cancer registries, which is also a very interesting topic. So I hope to see you all there in the next seminar. Thank you all for your time and for your effort.